Hello and welcome everyone. Sorry we had to end class early today, but I felt that was the safest thing to do with us all living in different towns and not really being aware of the safety matters that exist where everyone happens to be. I know in Mount Pleasant it was getting kind of nasty. So I just wanted to go over these activities with you so that you had the correct answers as you're studying for your exam. And so remember, in the setup of this question, we're talking about how this virus, hepatitis B, can cause liver cancer and how it's linked to P21. And so let's go over these questions here. And I went ahead and added some of your answers to these questions just so we could use those as a jumping off point to talk about these. So up here, the top one, this is what would normally happen. We get a mutation in the DNA. P53 recognizes that mutation, it activates P21. P21's job is to bind to CDK. When it binds to CDK, CDK can't bind to cyclin. If it can't bind to cyclin, it can't phosphorylate RB, releasing E2F. So in this state, with a mutation in the DNA here, P21 puts a stop on the cell cycle. Once the mutation is fixed, P21 will degrade, and once again, the cell cycle can move forward. Now, in this particular cancer, what happens is that P21 is mutated. I'm just going to put an X through here to show that it's mutated. It's not functional. Now, later on, we're going to find out it doesn't actually have a mutation in it, but we can safely assume, based upon the question here, that it's not functional. So P53 still detects the mutation, and P53 thinks it's doing its job. It attempts to activate P21, but if P21 can't make a protein, then P21 can't inhibit the formation of CDK cyclin shown here. If CDK cyclin can still do its job, it's going to add phosphate groups to the retinoblastoma protein. And remember, when that happens, that causes this conformational change, releasing E2F from the binds of retinoblastoma, and then E2F can go inside of the nucleus and activate the genes necessary to drive that cell cycle forward. So P53 thinks it's stopping the cell cycle, but in reality, the cell cycle is continuing, and this mutation, because it's not been fixed yet, will persist. And every time this cell divides now, this mutation will now be part of that genome. Then eventually what happens, because of this overgrowing cell population, enough mutations will accumulate and will lead to cancer. Question 2. Is P21 a tumor suppressor gene or a proto-oncogene? The correct answer is that P21 is a tumor suppressor gene. Let's go back to the previous slide. Remember, P21, its whole job is to inhibit CDK so it stops the cell cycle. And remember the cell cycle, right? We have our G1, our S, and our G2, and then of course our M phase. P21 puts a stop to this. It stops it right here. It doesn't get past the G1S. And so since it stops the cell cycle, we know it is a tumor suppressor gene. The question doesn't ask it, but we might as well talk about it here. E2F is the opposite. It drives the cell cycle forward, so it is an oncogene. In this case, it's an oncogene, right? Up here, when it's functioning normally, it's a proto-oncogene. But once it's gone rogue here, no longer being regulated, it is now an oncogene. But P21 itself is a tumor suppressor gene. Okay, so now let's talk about question 3. Now, this is based on a real study that when they started sequencing the genes in this cancer that was infected with the HBV virus, they discovered that there were no mutations in known genes. More specifically, the gene for P21, which we can draw down here, so here's our P21 gene, they were expecting to find some mutations in this gene, but they did not find any mutation. By looking at the gene itself, one would have predicted the gene should be expressed, but they knew it wasn't expressed. So then they started thinking about an epigenetic model. And there's a lot of ways that epigenetics could be involved in causing P21 not to be expressed, but we're going to focus on the methylation aspect of it. More specifically, the DNA methylation aspect of it. And so we know that the promoter region here, P21, if it becomes methylated, the gene itself cannot be expressed. So even though the sequence of P21 is perfectly fine, it's still not expressed because what they discovered was that the promoter was methylated. This here, having these methyl groups at that promoter, is equivalent to having P21 with a nonsense mutation at the very beginning of the gene. In this case, it's transcribing this gene and it's making a protein, but the protein it makes is going to be very tiny. and it's going to be non-functional. So up here causes a methylation problem, and the protein is not made because of that. Down here, the protein is made, but it's a smaller protein and it's non-functional. Functionally, it's equivalent. No P21 is being made. One involves epigenetics, this top one, and one involves a gene mutation, the bottom one. Okay, let's talk about question 4. And we're going to assume that the answer to question 3 is correct, that there is an epigenetic model that methylates the P21 promoter, and that somehow HBV, it should be a B and not a P, HBV provides an enzyme that adds methyls to the P21. So the model now is this. You have your P21 gene here that we had on the last slide, and that HBV, the virus, when it enters a cell, it makes an enzyme that adds the methyl groups to the promoter, thereby shutting off this gene. And then with this gene shut off, like we talked about in the first question, that prevents the cell cycle from stopping and mutations accumulate, and that will lead to cancer. Okay, now what happens is the researchers discover this mouse. And I know this will be a horrible picture of a mouse. My bad. Looks more like a computer mouse than a real mouse, I guess. But we'll put whiskers on it so it looks like a real mouse. Oh my gosh, that's amazing now. All right, so we have a mouse mutant here that when you infect it with HBV, there's no cancer. This mouse can be chronically infected with HBV and still no cancer. So there is some kind of a mutation in this mouse that reverses this effect. And once researchers realized that the virus was adding methyl groups to P21 that caused it to not be expressed, they reasoned that maybe what's happening in this mouse is that there is a gene that we'll call DNA methylase. And we know DNA methylase, what it does is it removes methyl groups. And they reasoned that one of these DNA methylase genes here, which we'll box off here as the gene, that's in this mutant mouse, is somehow overexpressed. So that it's making a lot of DNA methylase. And while it's making this DNA methylase, it can remove these methyl groups just as fast as HBV is placing them in the promoter. And as long as this DNA methylase is degrading these methyl groups, P21 can now then be turned on when it needs to be turned on to stop the cell cycle. So I'm going to say this again. HBV causes methylation of the P21 promoter. This mutant mouse here has an overexpressed DNA methylase gene. This overexpressed DNA methylase gene produces lots of this enzyme, DNA methylase, that degrades these methyl groups, allowing P21 to be turned on in order to stop the cell cycle and prevent mutations from accumulating and ultimately preventing cancer. So the correct answer here, after all that explanation, is that the DNA methylase gene here would be overexpressed, as I said down here, and then over here I say that it is an intergenic suppressor as opposed to an intragenic. So why is it an intergenic suppressor and not an intragenic? Remember, intergenic suppressors, I'll just draw an arrow here, these occur when the suppressor mutation, I abbreviated suppressor, so the suppressor mutation occurs in a different gene. So here, P21 is the gene that's causing the cancer, and the suppressor mutation is in a different gene, DNA methylase gene. So since they're in different genes, we call them intergenic. An intragenic suppressor mutation is when the suppressor mutation occurs on the same gene. So in this case, if this were an intragenic suppressor mutation, then the mutation would have occurred somewhere on this gene that reversed the cancer phenotype. All right, let's look at question 5. Essentially, gene therapy is adding an unmutated gene into a cell that has a non-functional gene. This should allow the cell to function better. In practice, gene therapy has lots of technical problems, but for the purposes of this question, we're going to assume those technical problems are not present. So this is a kind of a challenging question. So let's just think about tumor suppressor genes in general. So for tumor suppressor genes to be impacted and to lead to cancer, both alleles have to be mutated in such a way that they're non-functional. Maybe there's a mutation in the gene here, maybe there's a mutation in the promoter. Somehow or another, we know that these tumor suppressor genes, let me put TSG up top so we know what we're talking about. These tumor suppressor genes make no proteins. And we'll put no tumor suppressor gene proteins. And because of that, when these mutations occur, the cell cycle can continue on without check. That's why it leads to cancer. What gene therapy does is it takes a non-mutated tumor suppressor gene, and for this purpose here, let's go ahead and just put some X's here in the tumor suppressor gene, just so we know that what we're talking about here is a mutation of some sort. So with gene therapy, what happens is you place this gene, this tumor suppressor gene, typically in some kind of non-infectious virus, and place this inside the cells. What happens then is the tumor suppressor gene will integrate here. Sometimes it's in a different location, but let's just put it right here, okay? It's going to integrate here with this new copy without mutations. Okay, so I'm just going to kind of scratch these out here. So now we have this new tumor suppressor gene here without mutations. And remember, with tumor suppressor genes, you only need one tumor suppressor gene to be present. One of them can be mutated, that's fine, but as long as one is not mutated, it will be able to stop the cell cycle as it needs to be. So with this tumor suppressor gene coming in here, we now know that the gene can now be expressed. It will now make tumor suppressor gene protein. I'll abbreviate protein just for brevity. And as long as this tumor suppressor gene protein is being made, it will be able to stop the cell cycle. This one's still not being made, and we just ignore it. In most cases, as long as you're making this one, you'll be able to stop this cell cycle. So, technically speaking, gene therapy could fix this problem with P21. And that's the answer I was looking for. All right, the second part of the question is, would it work for proto-oncogenes that have been converted to oncogenes? So let's think about that. I've erased what we had there before, because let's think about E2F, which we said was a proto-oncogene. And proto-oncogenes have their effect because they are overexpressed by one of several mechanisms that we talked about. But something has happened, and we're getting too much E2F or too much activity, because maybe it's, in this case, not being bound to retinoblastoma protein. But whatever the case is, there's just too much E2F, too much active E2F. And so if in our infinite wisdom we decided we wanted to infect these cells with an E2F gene, what we would be doing is essentially making the problem worse. We already have too many active E2F proteins. If we add more E2F gene, we're just going to create even more active E2F. But in general, when we think about the possibility of gene therapy, it usually will not work in the case of converting oncogenes back into proto-oncogenes. So here were the take-homes for that 5-part question. Be very familiar with the proteins involved in cancer progression. No doubt you're going to have to talk about this pathway, so make sure you have it memorized. The pathway I'm talking about is that P53 to P21 to CDK cyclin to RB to E2F to driving the cell cycle forward. Make sure you can put those all together. Think about what happens if any one of these genes is mutated. Be able to identify which of these genes are tumor suppressor genes and which are proto-oncogenes. Be able to link this problem set back to previous topics like epigenetics or small interfering RNAs, the siRNAs. Know the basic idea of gene therapy. Please don't go off and learn the intricate details of gene therapy. Just know that gene therapy is the process of replacing a mutated gene with a gene that's not mutated. All that technical stuff we don't need to worry about in this class. Okay, this next activity we didn't have time to get to in class because we ended early, but I wanted to make sure you understand the basis of this question. And I'm using this disease here called Burkitt lymphoma as the starting point of this question. This is a very rare form of cancer, and it's a cancer of our immune system's B cells. There are many different forms of these lymphomas with different degrees of severity and prevalences, but one of the main forms is one that predominantly affects children in Africa. It is linked to this gene called c-Myc. c-Myc is a transcription factor that drives the cell cycle into the S phase. And just to kind of make our story complete, it is a gene that is activated by E2F. So c-Myc is one of those targets of E2F. We know that c-Myc is overexpressed in Burkitt lymphoma patients. We also know, as I said up here, but I didn't stress it, I don't think, is that c-Myc is a transcription factor. And so it's going to be really important in driving this cell cycle forward. So c-Myc in Burkitt lymphoma patients acts as a blank oncogene or tumor suppressor gene. There's a couple clues here as to why we are going to say that it is an oncogene. We know that it's overexpressed. because the problem tells us that c-Myc is found to be overexpressed. So that's a clue that this is going to be an oncogene. We also know that it's important in driving the cell cycle into S phase. So this is one of the gas pedals of the cell cycle. So this is definitely an oncogene. Then down here, this is more of a review question from your notes. But what are the 3 ways this could be overexpressed? I want to spend most of my time talking about one of the ways, but let's talk about the other 2 first. So the first way is amplification. And I'm going to just abbreviate it here as AMP. If you have the gene c-Myc here, and you amplify it, so now there are 2, maybe 3 additional copies of c-Myc, that will cause it to be overexpressed. More copies of c-Myc, higher expression of it, and that's going to drive the cell cycle. And that will cause the cell cycle to move faster than the cells need it to. The other is, it could be a missense mutation. So in this example here, maybe there's a missense mutation that changes one amino acid for another amino acid, and that change is enough to make this protein c-Myc active all the time. Maybe it's always able to bind to MAX, or maybe because MAX is its binding partner that activates these other genes. Or maybe now with this missense mutation, it can just bind to these target promoters all the time, with or without these other proteins involved. But missense mutations can change the shape of the protein to allow it to be active all the time, and that is a second way that proto-oncogenes can become oncogenes. The third way is by translocation, and I'm going to go to the next slide to talk about that. We actually see this in the Burkitt lymphoma patients. And so what we have here are 2 chromosomes, chromosome 8 and chromosome 14. Near the ends of each of these chromosomes, not at the telomeres, but near the ends, we have the c-Myc gene and we have the IGH gene. c-Myc is going to be expressed in order to drive the cell cycle forward. IGH is an immune system gene, and it's going to be expressed due to an immune response, such as you might see when someone is getting sick with something like cancer. So when the cell recognizes that there are mutations that need to be fixed so that cancer does not occur, c-Myc will be turned off. And it's turned off because of this promoter in front of it. The promoter is signal, we don't want to make any c-Myc, so it stops it. In order to battle the infection, the promoter in front of IGH is turned on. So this is what should happen. c-Myc is inhibited, cell cycle stops, IGH is turned on so that we can have the appropriate immune response. Now what happens sometimes is that c-Myc and IGH can have a translocation. And in this particular one, what happens is IGH moves over here and c-Myc moves over here. But the promoters don't move. Remember, when translocations occur due to chromosomal breaks, the chromosomal breaks don't look for promoters, and so it can cut right around the promoters. It happens fairly randomly. So what happens here is this promoter here is being signaled to turn off. The cell thinks, so to speak, that it's turning off c-Myc. But in reality, it's turning off IGH, which is going to affect the immune response. Over here, this promoter thinks, so to speak, that it's turning on the IGH gene, but it's not. It's turning on the c-Myc gene. And that's going to drive the cell cycle faster at a time when it should slow down so it could fix those mutations. So again, translocation places IGH in front of a promoter that is turned off. That impacts our immune response. c-Myc is placed in front of a promoter that is turned on at a time when it should not be turning on c-Myc, but it does. And so we end up with a weaker immune response and faster cell division, which is a double-edged sword when it comes to our body's ability to fight cancer. So let's go ahead and talk about this activity from day 14. Now remember, in day 14, what we said was that a trisomy of one given chromosome, where you usually have 2 and now you have 3, or a monosomy, where you have 2 chromosomes for one particular pair, and now you have 1. So that was a monosomy, this one up here is a trisome. We said that in autosomes, so everything here that I'm circling, if a trisomy or monosomy occurs in these, it's usually not good. It is usually lethal, and if the individuals survive, they are usually very sick and usually don't have a typical life expectancy. Again, this was for autosomes. However, we said that the sex chromosomes were different, these two down here. Usually we are either XX or XY. However, sex chromosome trisomies or monosomies do occur, and so some of them were the XO, like in Turner syndrome, XXY, like in Klinefelter syndrome. We also mentioned XXX, which is just called triplo X. And then we also talked about Jacob's syndrome, which is XYY. Now, all of these individuals have some phenotypes associated with them, but no severe phenotypes at all. Cognitively, they're fine. Physically, they're mostly fine. And they typically have the average life expectancy. So the question here asks, why is it that autosome trisomies or monosomies are lethal usually, but trisomies or monosomies in sex chromosomes are okay, typically fairly healthy? Now, if you'd like, you can hit pause and think about it, or you can just let it play on and I'll give you the answer here in just a few seconds. Remember that someone who is XX and someone who is XXX and someone who is XXY and someone who is XY and someone who is XYY are all equivalent as far as the X chromosome is concerned. All of these individuals, due to X inactivation, have all of their X chromosomes inactivated except for one of them. And because of that, the total X chromosomal content is the same. I accidentally erased all this, but one thing I did want to mention about somebody who is, say, XYY, because they do have an extra Y chromosome. But it's important to remember that look how big the X chromosome is here, and look how embarrassingly small the Y chromosome is here. The Y chromosome has very few genes. Of all the chromosomes, it has the fewest number of genes. So you could give a person 2 Y chromosomes, you could give them 3 Y chromosomes, 4 Y chromosomes, and it's not going to have much of an impact. They might have things like increased acne, so nothing really all that detrimental. However, it didn't stop some scientists from doing a study where they looked at people with Jacob's syndrome, and the question they asked is, are Jacob's syndrome people more likely to commit crime? So is there an increase in crime if you have XYY genotype? Initially, the conclusions were yes, more crime. However, one thing I want you to remember, and sometimes I think the general public doesn't quite get this, is that science is a slow, sometimes, and a self-correcting discipline. And so what happened was other investigators came along and looked at their study. And looking at their study and repeating it, they said these conclusions were wrong. And in reality, no. XYY does not lead to more crime. Individuals who have Jacob's syndrome are not more likely to be criminals. The reason this study was wrong, and I'm surprised nobody caught it when they were doing the peer review, is that they only looked at people in jail. So if you only look in places where there are people who have committed crimes and you find that there are lots of people there with XYY, that doesn't necessarily mean that they're there because they were XYY. And so when they did a much larger study looking at people outside of prison, and I think they also looked at people in prison as well, they discovered that nope, having an extra Y chromosome is not any indication that you're going to commit more crimes. But as you can imagine, this study, before it got refuted, did cause some concerns in the general populations, and then certainly we've come to this topic before, the topics of eugenics came up a lot, like, well, maybe we should sterilize these people, or maybe we should make sure they don't procreate, or all this other things that are both socially and ethically charged. Okay, so you can read these take-home points. I'm not going to read them to you. You can hit pause and read them. Now, I have this last activity, which at the end of it, you may want to call me a liar, but please don't, because I'm going to explain an exception to the rule I just told you. Remember, I just told you that somebody who is XXY or XO or XX or XY all express one X chromosome. And as such, that's why people who have aneuploids in XXY or XO are for the most part okay. Now, if that is true, why do we see specific phenotypes linked to Turner syndrome? I'm just going to put TS. And why do we see specific phenotypes with Klinefelter syndrome. I really put these in the wrong order because XO is Turner syndrome and XXY is Klinefelter syndrome, but you know what I mean. Why do we see phenotypes with them if what I'm telling you is that you only express one X chromosome in all of them? I would predict, that all 4 of these individuals, as far as the X chromosome is concerned, should be fine. You would expect the XY individual and the XXY individual to be phenotypically similar, right? And the XO and the XX also. So why is that? There are a few reasons out there, but the 2 main reasons I want to go over with you. The first one is an issue of timing. When you have this 2N diploid cell, the zygote, it will go through several divisions. And it's not until it gets to somewhere between the 8 and 16 cell division that X inactivation occurs. So it's possible, and there's some evidence to support this, that these early cell divisions, both X chromosomes are active, and that is providing that difference that we see between these aneuploids up top versus the euploids on the bottom. And the second reason is the one where you may want to call me a liar, but don't. I didn't lie. I just withheld an exception until now. Sometimes you have to do that. I apologize. Remember earlier, and when I say earlier, I mean like the last slide, I said that the X chromosome, all the X chromosomes except one were inactivated. And I said, or at least hinted at, that it was 100% inactivated. Even my cat is upset about what I'm about to tell you. But that's not true. The X inactivation is only about 70 to 75% inactive. There are some genes that remain active on the inactive chromosome. And so this is probably the largest reason why we do see a difference in phenotype of the XXY individual or the XO individual compared to the euploid. Now, it's still not drastic enough where it leads to the severe health problems that we see with the autosome aneuploids, but is enough of a difference to give us those fairly mild phenotypes that we see in Turner syndrome, Klinefelter syndrome, Jacob syndrome, triple X. Okay, so these are the 2 main reasons. And so here were the 2 main reasons again. X chromosome is only partially inactive. A small portion of the inactive chromosome still expresses some genes. And also this idea of timing, that during early embryogenesis, all available X chromosomes are active. All right, so let me know if you have any questions, and I'll be happy to help alleviate any questions or concerns you might have. All right, take care. See you later.